Good morning. It's February 19th, and this is your daily brief in science. Here's everything you need to know. A recent study has unveiled a crucial genetic factor in the development of spoken language among early humans. Researchers have identified the NOVA1 gene variant, specifically the I197V variant, as a significant contributor to the origins of speech. This variant is present in nearly all modern human genomes analyzed and is believed to have originated from an ancestral population in Africa, providing advantages in vocal communication. Published in Nature Communications on February 18, 2025, the study indicates that the I197V variant does not interfere with RNA connections related to neuronal development or motor control. By comparing the genomes of modern humans with those of Neanderthals and Denisovans, scientists confirmed the absence of the I197V variant in these close relatives, highlighting its unique role in human evolution. While Neanderthals and Denisovans possessed anatomical features conducive to speech, they did not carry the I197V variant found exclusively in modern humans. This ability to communicate through speech is thought to have offered early humans a significant advantage over other hominins, aiding in information sharing and coordination. Using CRISPR gene editing, researchers modified the NOVA1 protein in mice to reflect the human variant, resulting in changes to vocalizations, particularly in pitch and call patterns. The study found that the human NOVA1 variant fine-tunes RNA messages in brain regions linked to sound production, thereby influencing vocalization patterns. Dr. Robert Darnell, a co-author of the study, has been investigating NOVA1 since the early 1990s. He aims to enhance our understanding of human origins and develop treatments for speech-related disorders. Darnell hopes this research will facilitate the early detection of individuals who may benefit from speech and language interventions. Future research will delve into NOVA1's specific role in language and developmental disorders, as well as its interactions with brain regions involved in sound production. However, researchers caution against labeling NOVA1 as the sole human language gene, noting that other genes, such as FOXP2, also play significant roles in language development. Carl Zimmer, an award-winning science journalist, is set to release his new book, Airborne: The Hidden History of the Life We Breathe, which delves into the complex history of airborne diseases and the insights gained from the COVID-19 pandemic. The pandemic underscored the critical importance of understanding airborne transmission, particularly highlighted by the Skagit Valley Corral outbreak in March 2020, where 58 members contracted the virus during a rehearsal. Research revealed that the outbreak was due to airborne transmission, with an infected singer releasing tiny droplets that spread the virus without the need for coughing or sneezing. The confusion regarding COVID-19's airborne transmission was significant, with the World Health Organization taking 2 years to officially recognize this mode of transmission, despite existing research. Zimmer emphasizes the necessity for improved ventilation and air quality measures to mitigate airborne diseases, a crucial lesson reinforced by the pandemic experience. Historically, the understanding of disease transmission has evolved, with early proponents like the Wells couple suggesting that diseases could be spread via microdroplets, now known as aerosol particles. The concept of aerobiology has roots dating back to ancient Greece, where Hippocrates proposed that corrupted air could cause diseases, a notion that lingered for centuries. The acceptance of germ theory in the late 1800s largely sidelined the airborne transmission perspective, which has only recently regained prominence in light of the COVID-19 pandemic. Traditional public health approaches often focused on close contact, complicating the acknowledgement and response to the airborne nature of diseases like COVID-19. Zimmer advocates for a balanced understanding of our interaction with airborne life, recognizing both the risks posed by pathogens and the benefits from non-harmful microorganisms. His book, scheduled for release on February 25, 2025, aims to reshape our comprehension of the air we breathe and its implications for health. Notably, the World Health Organization declared COVID-19 no longer a public health emergency on May 5, 2023, marking over 3 years since the virus first emerged. Researchers at the University of Queensland have unveiled a groundbreaking technique that employs nanoparticles to deliver synthetic mRNA into plants through their roots. This advancement is poised to revolutionize rapid crop improvement, allowing for quicker and more efficient plant modifications compared to traditional breeding methods, which often take years or even decades. The mRNA introduced into the plants operates similarly to mRNA vaccines used in humans, producing proteins temporarily before naturally degrading. A key finding from this research is that the nanoparticles can distribute their genetic material throughout the plant via water movement, rather than being confined to the initial cell they entered. This ability to spread mRNA throughout the plant represents a significant breakthrough in plant genetic modification. The research team adapted a benign nanoparticle originally designed for vaccine and cancer treatment delivery in animals for this purpose. To penetrate the tough cell walls of plants, these nanoparticles are coated with a special protein that softens the walls, facilitating the delivery of genetic material. Professor Bernard Carroll noted that this technology could fine-tune plant genes to enhance crop yield and improve food quality. The implications of this research are substantial, as it could lead to the rapid development of new crop varieties that address specific issues like flavor and quality more swiftly than traditional methods. The University of Queensland has patented this innovative technology and is actively seeking partners to advance its application in real-world agriculture. The advancements from this study could significantly enhance food production methods, improving crop resilience and nutritional quality while tackling global food security challenges. The findings were published in the journal Nature Plants on January 2, 2025. A new clinical trial is reshaping the treatment landscape for localized muscle-invasive bladder cancer. The Immunopreserve trial, led by Dr. Xavier Garcia del Muro from the University of Barcelona, has produced promising results using a combination of radiation and immune checkpoint inhibitors, durvalumab and tremelimumab. This Phase 2 study involved 32 patients with stage T2 to T4 AMIBC who had not previously undergone immunotherapy or radiotherapy. They received 3 courses of the 2 immune checkpoint inhibitors alongside concurrent radiotherapy. Remarkably, among the 28 evaluable patients, 93% achieved a complete response. The estimated 2-year overall survival rate stood at 84%, with a distant metastasis-free survival rate of 83%. After a median follow-up of 27 months, 30 patients successfully preserved their bladders, while only 2 required radical cystectomy due to recurrence. The treatment regimen was generally well tolerated, though there was a 31% incidence of grade 3 or 4 adverse events and 1 treatment-related death. Currently, radical cystectomy is the standard treatment for localized MIBC, which is highly invasive and can significantly impact quality of life. Preclinical studies suggest that combining immune checkpoint inhibitors with radiotherapy may yield similar efficacy to chemotherapy, while reducing side effects. However, Dr. Garcia del Muro noted the need for larger studies to validate these findings and compare this bladder-preserving approach to standard treatments. The study does have limitations, including a small sample size, relatively short follow-up duration, and a lack of comparative data with traditional treatments. An alternative non-invasive strategy, involving transurethral resection of the tumor followed by chemotherapy and radiation, is also being explored, but comes with its own challenges. Overall, the Immunopreserve trial indicates a potential shift in treatment paradigms for localized MIBC, focusing on bladder preservation and enhancing patient quality of life. Astronomers at Brown University have made a breakthrough in tackling a persistent problem in radio astronomy, radio frequency interference, or RF1. As the number of satellite constellations has surged, surpassing 11,330 as of mid-2023, astronomers are facing significant challenges in obtaining clear radio observations. This research was sparked by unexpected detections of television broadcasts while analyzing data from the Murchison Wide Field Array in Western Australia, a designated radio quiet zone. Utilizing advanced techniques like near-field corrections and beamforming, researchers identified the source of the interference to an airplane flying at 38,400 feet, which was reflecting signals from Australian digital TV Channel 7. This finding supports a long-held theory that stray television broadcasts could bounce off aircraft, presenting a potential new method for filtering out RF1 that affects astronomical observations. Unlike traditional RFY management approaches that often lead to substantial data loss by discarding entire observations, the new technique focuses on isolating and eliminating only the interference sources. By identifying these RFI sources, astronomers can model and filter out the disruptive signals, thereby reducing data loss and enhancing the chances for significant discoveries in radio astronomy. Collaborations with organizations such as the National Science Foundation and SpaceX are in progress to develop real-time data-sharing systems aimed at mitigating satellite interference. Future efforts will concentrate on refining the filtering techniques to address the more complex challenge posed by the vast number of satellites currently in orbit. Pober highlighted that if Earth cannot provide a quiet sky for observations, alternative locations, including the Moon, may need to be considered for the construction of radio telescopes. The next steps will involve developing methods to remove similar signals from astronomical data, particularly those emitted by overhead satellites. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.